Good morning everyone. Thank you for taking the time to watch my video presentation about my research. Today, I'll be discussing my study titled Comparative Genomics of the Purple Land Crab, Gecarcoidea lalandii. My name is ZiweiWu, and I'm currently at the University of Melbourne. I'm excited to share with you some key findings from the research I conducted during my master's degree at Sun Yat-sen University. Our study centers around the unique adaptations that enable the purple land crab, Gecarcoidea lalandii, to thrive in terrestrial environments. I've divided my presentation into 4 parts to guide you through the research. The first part is the research background. As many of you are aware, the transition from marine to terrestrial habitats is a significant milestone in evolutionary history. However, as you can see in the slide, this transition poses 7 major challenges for organisms, including issues such as gravity, osmoregulation, air respiration, nitrogenous waste management, and others. These obstacles have spurred species diversification, but the molecular mechanisms that allow animals, like arthropods, to successfully adapt to life on land remain largely unknown. Our research zeroes in on Gecarcoidea lalandii, a pearl-color carp that lives in damp terrestrial burrows. By analyzing its genome, we aim to uncover the molecular mechanisms that have facilitated its successful adaptation to terrestrial environments. We combined short and long-read sequencing, high-C technologies, and transcriptomic data from 28 species to uncover key molecular mechanisms of terrestrial adaptation through comparative genomics and transcriptomics. We began by sequencing the genome of G. lalandii, achieving a highly accurate and complete assembly. The genome is 1.33 gigabits in size, with an N50 value of 21.25 gigabits, indicating its quality. We compared its genome to those of 8 other crustacean species and 1 insect species. This figure shows the phylogenetic relationships among these species. Through this comparative analysis, we identified both gene family expansions and contractions. Notably, we identified expansions in gene families involved in fatty acid synthesis, critical for energy storage and metabolism. This is especially important for terrestrial species, where maintaining body temperature and energy supply on land is more challenging. We also observed expansions in the PAX gene family, essential for visual neuron development, and the cadherin gene family, which is important for cytoskeletal organization, a key adaptation for physical environments. These expanded gene families play roles in important KEGG pathways, including steroid hormone biosynthesis, thermogenesis, and fatty acid metabolism, pathways essential for survival in terrestrial environments. Many of these pathways were found to be under intensified selection, suggesting that natural selection has optimized these genes for life on land. To further explore how gene expression changes during terrestrial adaptation, we analyzed the transcriptomic profiles of 28 crab species. We identified 63 genes under intensified positive selection in terrestrial species. These genes are linked to immune responses, growth regulation, and cell signaling, showing how land-based crabs have evolved to combat challenges like solar radiation and pathogen exposure. We also found instances of convergent molecular evolution in species that independently transitioned to land. Key pathways, like the nod-like receptor, WNT, and hedgehog signaling cascades, which regulate immune responses, tissue repair, and gill development, showed convergence, suggesting that species adapting to terrestrial environments share molecular strategies. In conclusion, our study offers valuable insights into the molecular mechanisms driving terrestrial adaptation in crustaceans. By combining comparative genomics and transcriptomics, we have deepened our understanding of the genetic basis for these adaptations and provided a foundation for future research, particularly in mariculture. Our findings have potential economic benefits, particularly in improving the environmental resilience of marine farms. Thank you for your attention. Please feel free to follow me on LinkedIn and Twitter if you have any questions.